Good evening, everyone. Class will start at 5:40. Everyone be online and be patient. Devdatta, please give co-host to madam.
Sir.
Okay, sir. Okay, sir.
Okay, madam.
సార్ నేను దీన్ని మూవ్ చేయలేనా?
చేయగలరు మేడం.
నాకు రాలేదు.
ఇది మీరా షేర్ చేసింది ఎవరు? మీరేనా?
నేను షేర్ చేయలేదు సార్. నేను షేర్ చేద్దామంటే నాకు అలో ఇవ్వలేదు.
ఇప్పుడు ఇస్తున్నారు. జస్ట్ డెవదత్తా యూ ఎగ్జిట్ స్క్రీన్ షేరింగ్, మేడం విల్ షేర్. మీరు ఫోన్లో మీరు చేసుకోవచ్చు కదా, మీరు చేయగలుగుతారు కదా?
పీడిఎఫ్ నేను చేయగలను సార్.
ఓకే, ఎందుకంటే మీరు స్క్రీన్ షేర్ చేస్తే మీకు మూవ్ చేయొచ్చు ఈజీగా. సార్ నెక్స్ట్ అనాలి. ఓకే
ఓకే సార్.
మేడం, నౌ యు ఆర్ కో-హోస్ట్, యూ కెన్ షేర్.
వచ్చింది సార్, ఆప్షన్ వచ్చింది సార్. Sir, now it is visible, sir?
Yeah.
Okay. Shall I continue, sir?
Wait, madam. We will start at 5:30, 5:40.
Okay, sir. I will wait.
Madam,
sir.
you are sharing from phone, na?
అవును సార్. సార్
Ma'am, try rotating your phone in a landscape mode.
Okay.
Yeah, it's fine now.
Okay.
Okay, ma'am.
Okay.
Sorry, actually, my lap is stuck. మేడం
సార్ చెప్పండి సార్.
అది మేము షేర్ చేస్తాము పిపిటి. ఇది హాఫ్ హాఫ్ కనిపిస్తుంది స్క్రీన్, అంత బ్లాక్ వస్తుంది.
ఓకే సార్, నాకు క్లియర్ గానే ఉంది.
అంటే ఇది వీడియోలో హాఫ్ వస్తుంది.
ఓకే ఓకే సార్.
వీడియోలో
సరే సార్, మీరు షేర్ చేయండి, ఇంకా నేను చెప్పినప్పుడు స్లైడ్స్ మారుస్తారు.
స్లైడ్ జస్ట్ మేము మూవ్ చేస్తా ఉంటాం.
ఓకే సార్.
ఓకే. దేవదత్త,
Yes, sir.
you are logged in from what, this lab or phone?
Lab, sir.
Okay, just you will share and madam will madam will be saying continue, then next then we will continue. Move.
Slide is looking okay only, sir.
Okay, na? For me it's looking half like that.
Okay, sir, okay.
Okay. Then okay, madam, you will continue.
Okay sir, shall I start?
You will start, madam, now. Okay, good evening, students. As we know that yesterday's class is postponed to technical region, and this is the last demo class and first regular class. So I am welcoming Dr. Reddyamini madam to continue today's session on mode of reproduction in crop plants. The basic thing, floral biology is one of the basic thing for JRF or SRF, whatever any plant science students. So madam, please continue.
Okay sir, thank you sir. Good evening to all. My name is P. Reddyamini. Today I am going to deal with a basic topic, that is modes of reproduction in crop plants, because this topic is very important for understanding the plant breeding principles and methods. In case of the plant breeding, the main aim is how to modify the genotype of the crop plants in order to make them useful to the human beings. In order to modify the crop plants or genotype of the crop plants, we have to understand how they are propagating, that is, how they are producing the new offspring. So if you understand this concept, then it will be easy for you to determine the breeding procedure and also which type of the genotype you have to develop through plant breeding methods in the respective crop species. That's why today I am dealing with modes of reproduction in crop plants. First, what is meant by reproduction? The method by which new individuals are produced from their parents is known as reproduction, and the manner in which they are originated
Madam,
from
just a minute. Madam, just a minute.
Sir?
Devdatta, live started or not?
Started,
Yes,
sir. Started.
Aditya.
Started,
Okay.
sir.
Continue, madam. In YouTube also. Yeah. Okay. Thank you.
Okay.
Madam, continue, please.
Okay, sir. The manner in which the new individuals are originating from their parents is called modes of reproduction, and these modes of reproduction is classified into two different types. The first one is asexual reproduction and sexual reproduction, and this asexual reproduction is again divided into two types: vegetative reproduction and apomixis. And this based on the flowers or plants involved in the reproduction process. This sexual reproduction is also divided into autogamy and allogamy. And this mode of reproduction helps in determining the genetic constitution of the crop plants. That is, whether the plant is homozygous or heterozygous will be determined by the mode of reproduction and mode of pollination. And if you know the mode of reproduction, it is easy for a breeder or a person who want to improve a particular crop for the deciding of the breeding procedure that has to be followed for the improvement of the respective crops. And this knowledge of the mode of reproduction is important for making the artificial hybrids, because the production of the hybrids between the selected parents or desirable parents is the basic step in almost all the plant breeding programs. And first you have to understand what are the vegetative parts and reproductive parts of the plant. Root, stem and leaves are the vegetative parts of the plants, and these are the flowers are the reproductive parts of the plants. These parts are involved in vegetative and sexual reproduction in the plants. The first type of the reproduction is asexual mode of reproduction. And in this type of the reproduction, there is no fusion of male and female gametes. And new plants, that is a progeny, will be developed either from the vegetative parts of the plants, that is through vegetative propagation, or it may arise, new progeny may arise from the embryos that are developed without fertilization, that is apomixis. These are the two basic methods that are involved in the asexual reproduction. The first one is vegetative reproduction. And in case of the vegetative reproduction, new plants arise from the different parts of the plant, that is through vegetative plants, parts of the plant, either through natural vegetative reproduction or through artificial vegetative reproduction. Coming to the natural vegetative reproduction, new plant develops from the portion of the plant body, either it may be a leaf or it may be a root or it may be a stem. And this new plant will arise when an axillary bud of the plant will develop into a lateral shoot. After that, this lateral shoot will develop into an individual plant after rooting. And those roots are called adventitious roots. If you separate those plants, those lateral shoots, and if you sow them, they will grow as a normal or individual plant. And thus, new plants may arise from the underground stems, subaerial stems, roots, and bulbils of the parent plant. The main advantage of the vegetative reproduction is the progeny that is produced is completely similar to that of the parental characters. And these are the different types of the vegetative reproductions, and stems of the plants are modified into underground or storage organs in some plants, and these underground stems are also used as a propagating materials in some crops. And these the birds that are present on the underground stems, they develop into the shoots and function as a plants after rooting. And these are the examples for the underground stem modifications. In case of the potato, tubers are the storage organs, and eyes present on the tubers are also used as a propagating material. Whereas in case of the bulb, stem is modified into a reduced disc-like structures, and on the stem, leaves are arranged in concentric circles, and the lateral buds that are arised from the leaves are used as a propagating material in case of the onion and garlic. Whereas in case of the ginger and turmeric, the stem is modified into a horizontal storage organ called rhizome. These are the new buds, the buds that are present on the rhizome, and these buds are developed into a new shoots after the formation of the roots. And in case of the corm also, stem is modified into a storage organ, and the lateral buds that arise from the that are developed from the corm are also used as a propagating material in these crops. And there are some subaerial stem modifications, and these modifications include stolon, runner, and suckers. And these are the, in case of the wild strawberry and date plum, these are the offshoots, and runners are used as a propagating material. And subaerial stems are also used as a propagated material in case of the mint and date palm. Another stem modification, another type of the modification is bulbils. In these bulbils, flowers are modified into a bulbil-like structure, and these bulbils can be used as a propagating material. And here there is no formation of the seeds. Directly, the bulbils are used as a propagating material, and these are the vegetative bodies because these bulbils are developed without the fusion of male and female gametes. So there is no involvement of the fertilization and seed formation. The bulbous development is commonly observed in garlic and agave. In case of the garlic, the lower flowers in the inflorescence are developed, modified into the bulbils, and these bulbils are used as a propagating material. And the scientists are conducting research to induce bulbil development in plantation crops also, and they succeeded in developing the new plants through bulbils in cardamom crop. And the next method is artificial vegetative propagation. So it is commonly used for the propagation of the many plant species, even though they undergo normally, even though they do not undergo vegetative propagation through nature. And we can modify any plant organ, and we can use those plant organs as a propagating material. In case of the sugarcane, grapes and roses, we can use the stem cuttings as a propagating material. And in case of the fruit trees and ornamental shrubs, layering, budding and grafting methods are commercially used for the propagation of these crops. And there are several techniques that are developed for the vegetative multiplication through tissue culture also. In many plant species, they have developed protocols for the development of the new plants through tissue culture, and they succeeded in many ornamental and fruit crops like banana. Even though in those crops there is a sexual reproduction process, but mostly we are preferring the asexual reproduction due to some reasons. And the another type of vegetative reproduction is apomixis. In case of the apomixis, we will see the development of the seed, but this seed is developed without the fusion of male and female gametes. And the genotypes that are developed from the apomictic plants are completely similar to that of the parental plants. First time the apomixis was developed by Leeuwenhoek in citrus seed. And based on the presence or absence of the apomixis, we can divide the crop plants into two types. First one is obligate apomixis plants. In those type of the plant species, plant can reproduce only through the apomixis method. There is no sexual reproduction method for the propagation of those crop plants. And the another type is facultative apomixis. In those species, sexual reproduction occurs along with the apomixis. That is, these type of the plants can multiply either through apomixis method or through sexual reproduction method. That's why these plants are called facultative apomictic plants. And another different type of the mode of the reproduction is sexual mode of the reproduction. In case of the plants, there are sexual reproductive organs called flowers. In those flowers, male and female reproductive organs are produced, and those organs results in the production of the male and female gametes. Fusion of male and female gametes leads to the production of the zygote, which further develops into an embryo, and this embryo further develops into a seed, and this seed is used as a So, in case of the sexual reproduction, the propagation of the plant will occur by the formation of the seed, which are developed through fertilization and And if you want to understand the sexual reproduction, first you have to know the information about the flower and flower plots. Flower is a modified vegetative bird, and it develops floral organs, and which contain the sexual reproductive organs, that is gynoecium and androecium. And there are 4 different floral parts. The outer whorl is calyx, and this calyx is represented by the sepals. And these sepals play an important role in protecting the bird flower during the bud condition. And mostly these sepals are green in color. And the next oral is calyx, and it is the second floral organ that is present in the flower. And this corolla is represented by the petals, and these petals are attractive in color because these are the major agents which are useful for the attracting the insects for pollination, either for the cross pollination. This attractive type of the corollas are observed mostly in cross-pollinated crop species because their mode of pollination is cross-pollination. So for favoring the cross-pollination, they develop the petals which are having attractive colors. And the next floral organ, these calyx and corolla are considered as the vegetative parts of the flower because they are not directly involved in the sexual reproduction process. And the next one is, next floral organ is androecium. And this is the male floral organ of the flower. It contains, this androecium is represented by the stamens. Stamen contains two parts, the anther and filament. In the case of the anther, pollen grains are produced, whereas the filament is the stalk of the stamen. And the next floral organ is gynoecium, and it is the female floral organ of the flower. And this gynoecium is represented by the carpels, and it contains three parts. First one is stigma, style, and ovary. And this stigma is apical part of the apical end of the style, and this stigma secretes stigmatic substances which will favor the germination of the pollen grains. And after that only these pollen grains will germinate and release the pollen tube. And the narrow elongated part of the pistil between the ovary and stigma is called style. And the ovary is the organ which bears the ovules of a flower. And these are gynoecium and androecium are the reproductive organs of the flower because they are involved in the production of the male and female gametes. And this sexual reproduction involves three important steps. The first one is porogenesis, and next one is gametogenesis, and third one is fertilization. And these are the three important steps involved in the formation of a seed. The first one is sporogenesis. In case of the sporogenesis, microspores and megaspores are produced in their respective floral organs because here the spores are produced. That's why this process is called sporogenesis. And in case of the microsporogenesis, microspores are produced in the anthers. That's why it is called as microsporogenesis. And whereas the megaspores are produced in the flower ovule, that is called as megasporogenesis. From these spores, megaspores or microspores, gametes are produced. That's why the next process is called gametogenesis. This is the complete life cycle of a plant. In case of the flower, the first from the male side, the microsporangium present in the anther contains many microspore mother cells. This microspore mother cell will undergo meiosis division, leading to the production of the microspores, and this process is called microsporogenesis. After that, this microspore further undergoes mitotic divisions, two mitotic divisions, leading into the production of a pollen grain which contain one tube cell and two male gametes or sperm nuclei. And this is the pollen grain which is used for the fertilization. And in case of the female side, the megaspores, that is megaspore mother cell, is developed from an archisporial cell, and this megaspore mother cell undergoes meiotic division, leading to the production of a linear, that is a tetrad of the 4 megaspores. Among these megaspores, upper 3 megaspores will degenerate and the lower one will become functional. This functional megaspore again undergoes 3 different nuclear divisions, that is mitotic division without cytokinesis, and leading into the production of 8 nucleated embryo sac. After the rearrangement of the nuclei in the embryo sac, it leading to the production of the egg cells and synergids, and 2 polar nuclei in the center and in the 3 antipodal cells. And the next step is fertilization. And this is the fusion of male and female gametes to form a zygote is called fertilization. And in this fertilization, the pollen grain which is germinated on the stigma will will pass through the style and reaches the embryo sac. After that, it bursts and releases the two sperm nuclei into the embryo sac to micropolar end. One of the sperm nuclei that is present in the pollen tube will fuses with the egg cell, leading into the project that is present in the pollen tube will fuses with the polar nuclei, that is secondary nuclei that is present in the embryo sac, leading into the production of endosperm, that is primary endosperm nucleus. This is called double fertilization process, and the embryo will further develop into a seed and ovary develops into the fruit, and this seed is used as a propagating material. Thank you. Sir, completed, sir.
Okay, madam. Thank you. Very nice session on basic floral biology. Okay, Devdatta, Aditya.
Yes,
Just
sir.
you will
doubts, if you have any doubts, you can ask.
Yeah, just you conclude. Ask if you any doubts and then conclude, and just introduce about our program. I am outside.
Okay, sir. Okay, sir.
Okay.
Students, if you have any query, you can unmute yourself now and ask the question.
Hello.
Yes. Yes, please pick up.
Ma'am, you didn't discuss about apomixis type, madam. I have a doubt in apomixis in aposporia and adventative embryony.
Okay, I will discuss in detail in apomixis in separate class. This is just a demo class, now that's why I had taken only basic part. So apomixis will be there in separate session.
Okay.
In separate class, I will discuss about the apomixis completely. There is one more class for apomixis. So
Madam,
then at that time you will understand.
Madam, just give brief about that. Just briefing about apomixis. Can you possible? Can you?
Okay. Apomixis means all of you know that apomixis is just the formation of the seed without fusion of the male and female gametes. In case of the adventative embryony, the cells, that is vegetative cells that are present on the ovule, that is nucellar cells or other cells, integument cells that are present in the ovule, in the ovary, they may be modified into a new seed without fertilization. So that is called adventative embryony. And there is one more type, that is parthenogenesis. The egg cell may be developed into a seed or embryo, so without fertilization. So if the egg cell is produced from the embryo sac without reduction division, the egg cell is diploid in nature. So the seed that is developed from that egg cell is also diploid in nature. That is called diploid parthenogenesis. And there is one more type, that is haploid parthenogenesis. In those haploid parthenogenesis, the egg cell that is developed from the reduced embryo sac is haploid in nature. This egg cell will further develop into an embryo, leading into the production of haploid plants.
ma'am, there is one question in chat. One student have asked. He asked, I don't know what is this. May we get to know about micropyle and chalaja?
So these are the
One question, Patel. If can you elaborate what is your question? Please unmute your mic and you can ask.
Just a second, sir. To add to the point of aposporia and adventative embryony, like one student asked, adventitious embryony and apospory both will start from somatic ovular tissue, right? And if the somatic cells directly develop embryo from the outer covering, integument cells mostly, that condition is called adventitious embryony. And if the somatic cell acts itself as a spore, that condition is called apospory. To summarize the thing in one sentence, this is the difference between the apospore and adventitious embryony. I think you are clear right now. Yes.
Hello.
Yeah, please.
Hello.
Yeah, please.
Ma'am, can you again say about apospore, ma'am?
Apospory is the condition where the somatic ovular tissue, somatic cell, will act as a spore itself. Okay, act as a spore. It will
Uh,
not. It will not form the embryo
okay,
sac.
ma'am.
It will itself form a spore. That condition is called apospory.
Ma'am, but you are saying about spore means sporophyte directly from a spore. But
I'm sorry,
in
your voice is not
apocyte,
clearly audible.
Piyush, you can type in chat. Your voice is not clear.
Yeah. Please. Don't worry. I'll
okay,
write
okay,
the
okay.
answers. We will write the answers in the chat box. Don't worry. Ma'am, please proceed with the next question, ma'am.
Okay.
I think what this Patil is asking, may we get to know about micropyle and chalaja?
Chelazal end means at those ends we will observe the synergids. And in case of the micropolar ends, we will observe the XL and synergids will be present at the micropolar end. At the chelazal end, we will observe the antipodal cells. So through micropolar end only, this pollen tube will enter the embryo sac.
Hope Akshat Patel, is it okay? I hope you got your answer. Okay, sir. Yeah, thank you. It's ma'am is your
Micropolar
teacher.
end.
I'm not breeder. Any questions, students? Any questions regarding this? I think probably this is very fundamental. I think, though I am not belongs to this subject, but very fundamental. Okay, I think there is no more questions. Yes, someone is talking but unable to hear. Name is Chinmay Sahu from OUAT. You can you can put your doubt in chat box. Anyhow, students, thank you for your patience and thank you, ma'am. Thank you for your valuable time and class.
Thank you.
Yeah, regarding with our academy proceedings, what are the classes, how we are going to teach, all the details. Just now I have put it in the chat, and also we have uploaded the respective PDF, which includes all the details regarding this academy. We are charging very minimal charges compared with other organizations. We are very less. Probably this academy was not started with a profit oriented. We are all a PhD graduates and scholars from different universities, and some are working as a assistant professors in different universities. Hope you can use our academy knowledge. Thank you very much. Yeah, I think it's hope you all make a wonderful session and wonderful life with our academy. Thank you very much, students. If you have any questions regarding with our academy, you can ask now. Hope almost everyone have registered. Most of the students have registered. Students will have joined, I think so, but students who have not registered can ask any queries regarding with this academy. And you can read, ma'am had put one, she had giving a very good explanation regarding with your apomixis concept. You can read in the chat. Okay, I think there is no more questions. Madam, not, I'm asking you. Devdatta ma'am,
Yes, sir.
I think there is no more questions.
Yes, sir. We
Okay,
can
then.
conclude. Is the thing clear? Apospory and adventitis embryoni. I hope it is clear from the explanation. Try to read it once and you can contact if you have any questions, any query regarding this. Okay? Yes, sir, we can conclude now.
Okay, ma'am. Thank you very much for all your patience. Once again, I'm saying we are always here to guide you. We will put all our effort. Hope we will combinedly we can improve our knowledge in agriculture in our respective subjects. Not only you students, you're coming with your very young minds. You have very new ideas. So even as a PhD scholars, we also can improve ourselves in the subject. So combinedly we develop a good knowledge. I think ACA is a Agri Challenge Academy is a good platform for everyone. For everyone, it is very minimum charges is there. You can no way you can get such a low cost. I think hope you can understand what is you can compare with our academy to other academy. Definitely there will be tremendous variations will be there. Though our academy was not so fancy when compared with others, but remember students, we are not a profit oriented organization. We are also scholars. We are also doing a research in different parts of India. So thank you very much, students. For all the details, I have uploaded the PDF. You can go through it. Thank you very much, student. Thank you very much, ma'am. And students, now you can leave the channel.